So this is an open discussion for the audience. I'd just like to start by trying to bridge all the way from Carl's talk this morning to what we just heard and hear the thoughts of any of the panelists to address the issue of, with the fantastic precision neuroscience tools being developed at the level of understanding molecular interactions, but in particular imaging physiological responses of networks of neurons that are done in the experimental animals and labs, and the needs we've heard about on the clinical side. What does anybody foresee in terms of actually bringing those together? Are we going to be able to do that kind of high resolution optical imaging in a living human brain in a patient at some point? And will it have validity and usefulness? Or is that just too far away? What do you all think about that? I'd appreciate any comments, input. Hold your mics close.
So I guess the question is access, and that's a big problem for the brain in general. So to get the optical technologies into the human, you somehow have to use those genetic tricks, and so you'd have to have a strong positive outcome that you're shooting for that would allow you. Currently, I guess the only ways we can do that is with viruses. So I think that's probably coming later. I think the first thing that will come, my sense is that there will be, we will have people putting in the genetic tools to turn on or turn off neurons. Because that's kind of the next stage of what we do now with deep brain stimulation. So there are people who are working on, trying to put, channel rhodopsins into dopamine neurons so you can turn them on or off, better deep brain stimulation. So I think that might be the first step that we see in people. In the technologies that are coming out that I think will go into people will be better tools to measure the electrical activity where you don't have to inject a gene, but better electrodes to be able to interact with hundreds of neurons at a time. So we now have, say, the Utah array, which is in patients with brain-computer interfaces, people also putting those into people who are in the intensive care unit to look at their activity over time. That's an easy move, and then there'll be better and better those type of techniques. And so that's my guess. I don't know if anybody else wants to throw anything out.
Other comments from the panel on that subject?
Well, today, many institutions have intraoperative MRI, and we will use intraoperative MRI to help facilitate resections of tumors, for example, and that can include molecular imaging in the future, and certainly we do things like for infiltrative GBMs, glioblastoma multiformes, trying to determine what is tumor, especially when it gets to be a lower grade on the margins or whatever. What is tumor versus what is not tumor can be challenging, and there are molecular ligands that you can inject, and it's still in most places in a research phase, but you can inject and try to then, for us as surgeons, better determine what's abnormal and what isn't. So we're already utilizing that type of problem. The challenge with an intraoperative MRI is it's very expensive and it's frankly very time consuming. So even though you can get the time down, it cuts into the efficiency of patient care. So a lot of institutions have decided not to get it because it doesn't pay for itself. Other people, when they get it, they find they don't use it after the initial excitement over it, except for very unique, specific cases. So to be usable in the clinic, it has to be cost effective and efficient so it doesn't affect the workflow or have such a huge change in outcome that it's worth all those changes.
So, unfortunately, I'm a neurosurgeon, so I'm tragically concrete in my thinking. So let's think about some specific examples. So a patient comes in with a stroke, and maybe you can serially image them and get some incredible molecular imaging about the ischemic areas getting larger, or the hematomas getting bigger. Or longer term, somebody who may have Alzheimer's, you can see progressive accumulation of fibular tangles and other things, or Parkinson's or things. No question that better imaging will open up our eyes as to the progression of diseases, but I think Walter hit on a key point. What are you going to do about it? We need to concurrently develop techniques to either target genetic therapies or other ways to try to mitigate any adverse events that we see happening, and not only stop them, but even reverse them.
I think we still need to know more what's going on a basic level before we can utilize this very efficiently in the clinical setting. It comes to understand the different regions of the brain, not as some of you said this morning, not only on a local in a local area, but how the circuitry plays out, how the different parts of the brain interact, and how the neuronal circuits function. And this is incredibly complex, and I think we need to develop this in a stepwise manner by flies and mice and eventually on men. So this is a long-term process, but we need to start working on this. To use your image, we need to know how will we be able to put a man on moon. And instead of going to Mars, as I said, we should put our efforts into this to understand better how the brain works, especially the more complex cognitive functions, because that's what makes us human, as different from animals. And so how do we decide, and I'm looking to our colleagues here also at VTCRI, how do we decide and how do we imagine the future, and how can we stop impulsivity, step back from making wrong decisions, especially in the traumatized brain, because we see that these patients, they are violent, they're aggressive, they make bad decisions about their lives onwards. So they need, and we also see that the patients that have a good family networks, they do fare far better than the patients that have no networks and are left on their own. They end up in the streets oftentimes.
So maybe if I could, I want to put Carl on the spot a little bit, and I'd like to ask the question of the clinicians. If you had the resources of the Howard Hughes Medical Institute behind you, and I'll ask Carl to speak on behalf of them, and could ask for tools to be developed that you see having a need for in the clinic that could be game changers with the kind of expertise and sophistication that you have at HHMI and across the country, but use that as an example with Carl being here. What would be your dream machine they would make for clinical neurosurgery, neurology right now?
You're asking me?
Well, I'm asking them to ask you, and then I'm asking
I
you,
see. Okay.
you to answer and say how you can do it.
I
But go ahead.
quickly pick up on this theme. I mean, I think that from my perspective, it seems to me that there are many, many fundamental issues that we don't understand about both neural networks, but even the functioning of individual neurons. I think you can sort of take cancer research a bit as a guide, perhaps. In the 70s, there was the war on cancer, which I think many people considered to be a failure a bit, because we did not understand aspects about the cell cycle, regulation of cell growth, and it required really a push in really thinking about the basic biology of cell growth. And really now, 20, 30 years later, most of the drugs that are really rationally designed in the clinic that target cancers, there's been a lot of success for specific types of cancers. The war is not won, but many battles have been won, are really the result of a fundamental understanding of signal transduction, control of growth, and so on and so forth. And I really feel we're at this 1970s stage. Some of our lack of understanding is simply embarrassing. I mean, we do not know where channels are in neurons. We know this for few neurons, but channels make neurons work. We don't know where they are. We don't know where G protein coupled receptors are. We are now only starting to do a sequencing of neurons. This gives us a parts list of some of the the projects that Walter is spearheading at through the Brain Initiative, we get sort of a parts list of neurons and then figure out what they express in terms of transcripts, but we need a whole new revolution in localizing proteins in neurons. That will ultimately, you do this in models of disorders, then you probably get targets. You get probably very specific targets to deal with injury, ischemia, all these kinds of things that we really don't understand that well. That's just my opinion. I think there's still a really, really big role to play for basic research. But I'm happy to be bombarded on this other topic. So.
Yeah, I would agree that, as Carol mentioned, as we apply these technologies, you're talking about diseases, as we apply these technology disease models, that's going to open doors to new targets and better understanding. And so the thing I'd say is that a lot of the work now has been done in mice, but there are groups that are moving into non-human primates. So where I think the circuit dynamics will be kind of much more relevant to kind of behaviors we talked about. The other thing I mentioned is that one other avenue of progress, I think, is going to be exploiting the signals that we can get from outside the brain with a better understanding of when we match them up with the circuit activity we can measure inside. So electrical activity, magnetic encephalography, functional MRI, with the technology we now have to look at neural activity, we can build a better bridge between neural activity, the signals that give rise to those measurements that we can make, and we might be able to really refine our understanding of human brain circuits that way as well.
I think Walter made the comment that we used to look at anatomy and then try to put our therapies to correct that anatomy, and now the big area in the middle is the circuitry, which we don't understand as well. So the answer to your question, Mike, 5 years ago for me would have been, what I want to do is take much more than just the resources Howard Hughes has, but because it's going to be a bigger thing, but allow me to have a patient walk in the room with a problem, run a scan on them, and then I can turn around and walk into a hologram of that patient's brain and recognize immediately what circuitry, what issues are not working right, and say that's where we got to hit, and this is how we're going to fix it. But it's much more complex than that, because we got the circuitry, we've got the resonance of the circuitry, we got all those things. So it's more than just anatomy. It's how the anatomy functions and works as well. So kind of as we were talking about last night, this is a very, very complex thing, and what we want to do ultimately to apply to clinical practical uses is to get to the simplicity on the other side of complexity. And that's going to require the computational analysis that we've all kind of hinted at today.
Go ahead. Sure.
Just a quick comment. When it comes to the readout of the brain activity, I think that's very important, and it can help, especially people with a more intact brain but spinal cord injuries, to have artificial limbs and prostheses. So help them perform functions of everyday life in a better way that they can today. But that would be more difficult, I imagine, when the brain is scrambled by a traumatic brain injury. But anyway, that's also an avenue we should pursue.
I think I'd like to open up the floor for questions of the panel from anybody. Audra, go ahead, stand up and speak loudly, please.
Hi, Audra Van Wert, Virginia Tech. I have more of a philosophical question. So clearly there's great therapeutic potential for being able to stimulate deep in the brain, but you also worry that we're creating potentially addictive technologies, and we have people do-it-yourselfers creating transcranial magnetic stimulation devices in a garage. So what do you think about that, and should we take care? How do you think we should address that moving forward as we develop these technologies that have all this potential?
Well, that's already happening, and maybe not so much in the brain, but if you hang around athletes, especially high level, professional or top ranked college, they're always trying to get an edge, and they're always jumping into these quasi-experimental things. You can also go to dozens or hundreds of clinics, both in this country and elsewhere, to get stem cells injected for whatever you want. It's not very well regulated. So I understand your question, but I'm thinking that with time, the FDA will work through its processes and legislatures work through their processes so that you won't be able to have someone, in their garage putting in a, deep brain stimulator or something in you.
Yeah, I would just say that from the beginning at NIH, we've been very interested in neuroethical issues that are related. So you mentioned at the beginning that your thoughts are your own. That's no longer the case. I mean, they can put you in an MRI scan and they can tell what you're looking at from the readout of your brain. So the ethical issues that are coming out of the Brain Initiative are profound. So both the Europeans and the Americans have neuroethics as part of the Brain Initiative trying to approach these questions. But they're really going to be things that society is going to have to percolate with for a while before there's a light at the end of the tunnel. Because some of these are quite interesting. Yeah.
Part of it is our responsibility as healthcare professionals to help guide our patients, because a whole industry, as you said, is going to come out, and it's going to come out in brain health. Okay, Luminosity was just sued because they were making claims that they couldn't back up. There are people who want to do good, and you don't want to stifle innovation, but it has to be done in a way where you don't have people who are truly desperate for solutions become prey to charlatanism when it's not really proven and work. So that's kind of part of our responsibility as healthcare professionals, to help guide our patients in things that are proven. When it's not proven, make sure it's done in a appropriate research setting. And I actually see things like you have gyms right now for your health. I see cash payment for various brain health things coming out, and that's where the people are prone to charlatanism.
Questions? Yes, Greg, could you stand up?
Gregory, Virginia Tech. One of the interesting dilemmas that I think I face is that when we try to understand really complex behavior, we sometimes try to build it up out of relatively simple elements, and then the complexity emerges out of the interaction of those elements. On the other hand, much of what I find interesting about human behavior is about thinking and valuing and operating in really complex situations, particularly around decision making and action. And so I'd like to hear your take on whether it is likely that we're going to need to move to some complex, everyday human behavior as an analog, or not an analog, but as a complementary perspective on what we're doing in the slow buildup of understanding basic mechanisms in order to get to where I think we all want to go. How do we really understand the way that, and Tor, you said it so beautifully, when we are making ethical and other decisions about major issues that we as a society face. And it's not either or that I'm asking you about, but is there a way to think about going to more complex, real life, real human being systems that could be productive?
Well, I mean, just put in my 2 cents worth. I mean, there are, of course, decision making and value judgment, sort of in the sense of neuroeconomics. All kinds of animals do this. This is also has another name in a different literature called foraging and foraging theory. And it's remarkable that very similar principles and probably very ancient neural mechanisms underlie many of the basic components of behavior. Of course, studying ethics in a mouse is challenging, but short-term memory, how sensory information is evaluated to make one choice or another, those are the kinds of things, these elemental components that can be studied in model system, and if really understanding the neural mechanism is the goal, probably should be studied in model system. Then there is the challenge, right? How do you connect it to our own experience, our own inner lives, and to human behavior, and really bridging these gaps is a very big challenge, but that's something we really have to figure out how to do. How do we, and this is becoming possible. So for example, there's this phenomenon called the Bereitschaftspotential. You can, going back 30, 40 years, and you can read out based on EEG measurements, simple actions in a person before that person themselves is aware that she will make an action. And this is a classic neurophysiological behavioral phenomenon that now is really being deciphered in great detail at the cellular and circuit basis, for example, in rodents and primates and so on and so forth. So these links are being made, but it's a big challenge.
Yeah, I think, what I see is I see the scale up, probably we talk about scale, temporal, spatial, but there's also the behavioral scale. So people are looking at mice on these, the balls and the virtual reality, it's a very confined behavior. But then, and even in the animals, to kind of move into the whole spectrum of behavior and understand each of those is something that is going to be happening. And I think that's, again, where a lot of the countries are moving, Japan, China, and some folks in the U.S. are moving to non-human primates to enable that kind of work to be done with the same tools we have for mice, but now go into non-human primates and start to expand the behaviors. Now with people, there are going to be people now who have electrodes in for deep brain stimulation. Previously they were only for stimulation, but now they're for recording and stimulation. So there are people going to be walking around with constant electrical activity being recorded from their brains with all the behaviors that they do. Which brings up some of the ethical issues. Some of your behaviors, you don't want anybody to know what you're thinking when you're doing them. So I think that the data is going to be coming, and I think people have different scientific views. Some people are mostly interested in kind of the high-level human behaviors. They'll be using whatever tools they can. Some people at the very kind of fine control details. I know Reed Montague is really a great scientist in that zone who works out of here, could probably answer better.
Okay, I think we're going to have to wrap up. We're a little over time. Please join me in thanking all the speakers and the panel for this morning's session.